right, so we are going to talk about the nucleus and the nuclear membrane and the nucleolus today and how their structures relate to their functions. So the nucleus, structurally, let's go ahead and look at a picture of the nucleus. And so the nucleus is generally circular, but the big thing is that it just has a lot of room inside. It has a nucleolus, that's about the only set structure in there. We'll talk about it in just a second, but there's a lot of space in there for the chromatin, for the DNA. It is surrounded by its own membrane that is going to protect it. So the function of that membrane, we'll go ahead and talk about that now. It is a lipid bilayer, just like the cell membrane. It does control what enters and exits. So it does have that the hydrophilic outsides and the hydrophobic core. And it does have pores, so the nucleus can get things in and out that are too big to go through the membrane. And so that structure of that membrane is such that it is going to control what enters and exits the cell. Just like the cell membrane does for the whole cell. What enters and exits the nucleus, sorry, just like the cell membrane does for the whole cell. And so those pores are really important. One of the things that's going to need to leave that pore is your RNA, your mRNAs, your tRNAs, all of that stuff. So when DNA is transcribed into RNA, that RNA will leave through the pore out into the cytoplasm to go to the ribosome for translation. So that's the nuclear envelope or nuclear membrane, and how its structure relates to its function. The nucleolus is on the inside. The nucleolus is a structure that makes ribosomes. And so ribosomes are made of two things, rRNA and protein. The rRNA is going to be transcribed right here from the DNA, just like mRNA is. And so it is going to be transcribed here, and it's just going to go right into the nucleolus. The proteins are going to come from mRNA, so that mRNA is going to leave the nucleus through a pore. It's going to go out to the ribosomes. The ribosomes are going to make the protein. The protein will then re-enter the nucleus, go into the nucleolus, and then the nucleolus will package that rRNA and that protein into the ribosomes, which will then leave the nucleus again to go do their job of translation out in the cytoplasm or on the rough ER. And so that nucleolus, structurally, it is located so that it is accessible to the rRNA and the proteins, and it's also able to ship out those ribosomes. The nucleus itself, again, structurally, it is surrounded by that membrane, so it is protected. It is, I don't want to say empty because it's not really the right word. Spacious is maybe a good word. There's a lot of space in there for that DNA, because DNA is going to coil up and uncoil as it is transcribed or as it is not being transcribed. And so there needs to be space in there for that. So also replication is occurring in the nucleus during the S phase of mitosis, so that when the cell divides, both will have copies of the chromosomes. So spaciousness is a big thing in the nucleus. It allows for all of the functions that need to happen, transcription and replication. It is also closely related or closely aligned with the nucleolus because it's making rRNA that it goes into the nucleolus. And then, of course, the nuclear membrane with its structure and function. So I hope that helps. If you have any other questions, this is just kind of the words for all of that I just said. If you have any other questions about the nucleus, the nuclear membrane, also known as the nuclear envelope, or the nucleolus, if you will just let me know and we will go over those. I hope that helps.